Hello, I'm Tom Laver. I work with the University of Exeter Medical School. I've been asked to record this video to talk about how genetic risk can cause different forms of disease, and specifically what the difference between monogenic and polygenic risk factors are. So in this talk, I'm going to be talking about how genetic risk can cause disease in general, and again, specifically explaining what a monogenic risk factor is compared to a polygenic form of genetic risk. And I'm going to use diabetes as an example of a disease where you have both monogenic and polygenic forms of the disease. So starting off with how can genetic risk cause disease? So this plot is trying to show the extreme ends of whether your risk for a disease is entirely environmental, or whether your risk for a disease is entirely due to genetic risk, or as the case of many common diseases, whether it's a bit of both. So I've used the example of getting hit by a bus as an example of where you end up in hospital entirely by environmental risk, because the other examples people often give are things like infectious disease, which are obviously mostly environmental, but your chance of getting infectious disease and how you respond to it is also linked to your immune system, which in itself does have some genetic underpinning. So extreme example of purely environmental, getting hit by a bus, doesn't matter what your genetics are. You can be an Olympic runner, you're still not going to escape that bus. It's down to the environment. We go the other extreme of this. The classic view, at least, of diseases like Huntington's are you have a particular single genetic risk, and the entirety of you getting that disease is due to that risk. Whereas common disease, heart diseases, type 2 diabetes, or basically all forms of common disease, including most cancers, you have some genetic predisposition, but whether you actually get the disease or not depends on your environment. So I've talked about genetic risk. What do I mean by that? People often talk about having the gene for something, but this isn't, strictly speaking, the right language, because in almost all cases, everyone has all 20-odd thousand genes, and whether you have the disease or not, you still have that gene. What people actually mean is, do you have the variant of the gene that causes disease? Do you have the genetic change that are important for causing disease? So to use a kind of metaphor here, the human genome can be regarded as like a book. In simple terms, think of it as like a recipe book. It tells your body how to make proteins that you need to build your body to work healthily. And these genetic variants are changing the DNA sequence, and they're altering that version of the gene that you have. And if it's causing disease, that alteration is because it's causing it to work worse. So again, to use a metaphor here, if you had the instruction, ice the cake with pink icing, if then a mistake occurs in that instruction and it becomes ice the cake with pork icing, then that's obviously going to produce a rather different cake. Now, not all genetic changes are necessarily bad, and I'm sure there are some people who would regard pork icing as an improvement, but the idea being it alters the way things work, and most alterations are going to be bad. They're going to do something negative because of the way evolution has shaped your genes to be optimally functioning. If you disable the function of that protein, it's normally going to cause a problem likened to being a cause of disease. So talking about the extreme forms of genetic risk for disease, these are called monogenic forms, mono being one, so they are single genetic changes in key genes for that condition. So if we stick to our theme of diabetes here, like it could be a gene that affects insulin secretion or pancreatic development, something fundamental to your body's ability to process sugars correctly. So you have a variant in that severely damages the function of one of those genes that are really important for that pathway, and that alone is largely responsible for your development of diabetes. So again, to take our recipe book kind of metaphor, you have the healthy human DNA, you have a nice book, all the pages. Patient with a monogenic disease is not just you could regard it as not just a spelling mistake, but like the effect is fundamental. They've lost some important chapter, they've lost a page in this recipe. There's something fundamentally broken in their DNA that leads to disease, largely just due to that factor. In contrast, the common forms of disease are polygenic, poly being many. So polygenic diseases have lots of different genetic risk factors, each of them only conveying a small amount of risk. these then combine with environmental risk factors to decide whether you're going to get that disease. So in the case of type 2 diabetes, which is a very common disease, different people will have different levels of genetic risk, but some people at high genetic risk will still not develop diabetes, and other people who would be at low level of genetic risk for type 2 diabetes still develop it because of the lifestyle factors. So if you're severely obese, if you eat a high sugar diet, if you don't do any exercise, all these sort of things are in many cases a more important risk than your genetic risk of the disease, but it still plays a role. Using the book metaphor again, that perhaps you could think of the polygenic disease as a series of small spelling mistakes. They're all altering the function slightly, and they're having a small negative effect. But each single one, you can probably still understand the book, you can still follow the recipe. It won't necessarily make a difference overall, but each one makes things slightly worse, slightly more confusing, slightly more likely to cause disease, and then add that to your environmental risk. Now, this graph is a bit more technical than the rest of this talk, but there's a really important point I want to get across with it, which is that the frequency of a genetic variant in the population is very closely tied to how damaging it is, how likely it is to cause disease. So what this graph is on the y-axis, you have penetrance, which is the chance of having the variant causing a disease. You can think of it also as like effect, or how damaging a variant is, how serious the variant is, any of those sort of things. Whereas on the x-axis, you have how common the variant is. So in the top left here, you have what was labelled as Mendelian disease, Mendelian following a classic inheritance pattern established by Mendel, is the same thing as saying rare or monogenic. And now these variants have a really big effect, and that means they're really rare in the population because they've been heavily selected against by evolution. Whereas the bottom right-hand ones are your common disease variants. They have a very small effect individually, and that means they can be quite common in the population because people can have them without any particularly strong negative effects, so they're not heavily selected in the population. You will see very rarely some variants in the top right, which have a very big effect and are quite common, but this is only possible for some very specific strange cases. For example, some of the anemia variants in Africa that predispose you, protect you against developing infectious disease, they have a reason why they're able to be kept in the population at higher frequency, because they offer a beneficial effect while also causing potentially damaging effects. But you need some reason to be maintained in the population at a high frequency if you're having a big effect. Some interesting variants in the middle of this graph, which are relatively uncommon and having an intermediate effect. And this is partly due to the kind of our understanding of how disease work. It's very easy in some ways to find variants that are common with small effects, or really big but are rare. The ones in the middle are much harder to establish what effect they're having and how they work. But I think increasingly we are discovering they're also an important cause of disease and can contribute. Trying to put all of that together into a kind of a more simplistic formula, one way to think about this is the threshold concept. So you're imagining each person's individual risk is a bucket, and once you reach the threshold line, that is the point at which you have sufficient risk in order to develop the disease. Now, that risk might be a single genetic variant, it might be many polygenic variants, it might be environmental, and different people will have different causes for their disease. And also some people will be at some risk but won't go as far as developing the disease. So if you look at the examples I've shown here, individual A is the classic idea of a monogenic variant, a single variant that's causing a severe effect and is enough by itself to take you over the risk threshold and develop the disease. Individual B is more how we think of common diseases. They have multiple different risks. One of those might be polygenic risk, other might be, say, your exercise, another could be your diet. And cumulatively, those different risks are enough to take you over the threshold and cause disease. Individual C is still at risk. Maybe they have some genetic risk, maybe they have an environmental risk, but cumulatively they haven't had enough risk in order to develop the disease because they haven't reached the threshold yet, so they remain disease-free. But it's worth thinking about how this kind of concept ties together the different types of genetic risk factor working together and working with environment, and which ones are by themselves largely enough to cause disease and which ones require some more environmental impact. And that's the big difference between a monogenic disease-causing variant and a polygenic risk variant. And to talk about this in the context of diabetes and the different ways diabetes can be caused, there are a couple of different major forms of monogenic diabetes, the first of which is neonatal diabetes, which occurs almost exclusively in the first 6 months of life, which is a really severe, really early onset disease, which is typically caused by genetic changes that affect pancreatic development. Then there is a later onset form, which is arguably unhelpfully called maturity onset diabetes of the young. This is typically about 25 years of age people get diabetes, though it can be a bit earlier, and it is known to be, in rare cases, much later. Again, single genetic changes in important genes regulating insulin secretion. And then there's also two different major forms of polygenic diabetes. So type 1 diabetes is typically developed early on in life, but can also occur much later. It's autoimmune, where your own immune system attacks your insulin-producing cells, which is what leads to diabetes. But there's quite a lot of genetic risk around that, about whether you're likely to have an immune system that wants to attack your own cells. So those risk factors are polygenic. Again, there's no single large risk factor. There's several working together to increase your risk of whether you're going to develop type 1 diabetes. But exactly when you develop it will depend on when that autoimmune attack is triggered, which I think the best theory currently of when it's triggered is probably an over-response to a viral infection. The last type of diabetes is the most common type, which is type 2. Now, this is largely due to environmental risk factors, lifestyle, so again, obesity, diet, sugar level, exercise, those kind of things. They play the major role whether you're going to develop type 2 diabetes, but there are also, at this point, hundreds of small polygenic risk factors known to contribute to your risk, and those add together with your environmental risk to decide when you're going to get it. But it is largely a disease of old age and obesity, in contrast to the monogenic forms, which are much earlier. Why does any of this matter, is the point I'd like to end on. So there's the kind of blue sky point that science in general should be trying to improve our knowledge of how the human body works, how we get disease, and I, at least as a scientist, think there is some genuine value in just increasing the pool of our knowledge. But there's also a lot of very specific practical benefits that come from knowing the genetic causes of disease. So one of which is drug targets. So in the case of monogenic forms, where it's a single key gene is being affected, understanding which gene is affected and which genes are involved in this pathway, it can allow you to develop drugs that can help the people with those rare diseases to correct the genetic problem they have. But those drug targets can also be relevant for other forms of more common forms of the disease, because they highlight really key bits in the pathway that control, say, insulin secretion in diabetes. So maybe they can also help more common forms of disease if you develop drugs that target those. So I think it's really important for both those elements. Again, I've highlighted identify new genetic subtypes. This primarily affects monogenic forms, which is where you work out exactly the genetic cause, and that can lead to understanding better either treatment or management of the condition. So knowing when they're going to develop diabetes, knowing if they're going to develop any other non-diabetes features, which drugs are likely to work better on them. It allows much more personalized medicine to the genetic cause of their diabetes. And again, this applies to some extent in more common polygenic forms of disease, but that's more about estimating risk. So you can identify high-risk groups that are, based on their polygenic risk factors, more likely to develop a disease, say type 2 or type 1 diabetes, and that might allow you to monitor it, it might allow you to think about prevention, it might allow you to counsel for risk. But it's slightly less clear-cut than it is in the monogenic to say someone is going to develop, but you can at least identify a certain amount of risk and potentially do interventions. And again, that's a kind of an area medicine is moving into. The whole concept is precision or personalized medicine. How can we treat people based on their characteristics? And those characteristics can and should include genetic risk for disease. So a very quick summary of all the points I've talked about. Monogenic disease is caused by key variants, single variants disrupting key genes in pathways that regulate the kind of the fundamental pathways. So in diabetes, that would be insulin secretion, pancreatic development, genes involved in those. Polygenic is lots of different risk factors, each of which only has a small risk, add up and contribute to your risk of the disease, but it still relies on a lot of environmental and lifestyle factors to decide if you're actually going to get the disease. And in the context of diabetes, there are both monogenic, rare forms of disease, and most cases of diabetes are polygenic, whether that is the autoimmune type 1 or the much more common type 2 diabetes, which is more of a disease of old age and obesity. And I tried to pitch to you at the end why this matters, why understanding the genetic causes of disease is important, because it improves our understanding of fundamental biological pathways, but could also lead to better treatment for those people and potentially lead us into precision medicine, improving healthcare for everyone, which I think is kind of broadly what we should all be aiming for. And I hope after this talk you understand a little bit more about how genetic risk factors can contribute to development of disease. Thank you for listening.